Different types of staining procedures. Why stain bacteria? In their natural state, most of the cells and microorganisms that we observe under the microscope lack color and contrast. This makes it difficult to detect important cellular structures and their distinguishing characteristics. Certain techniques involving stains and fluorescent dyes have been developed to identify specific microbes, cellular structures, and DNA sequences. Staining bacteria helps us figure out the cell morphology, the cell wall structure, extracellular characteristics, and aids in identification of unknown microorganisms. Staining procedures take place in laboratories or clinical settings. Key terms. A smear. A good bacterial smear will be thinly visible to the naked eye. Too many cells means that the smear is too thick, which causes the slide to stain unevenly and may lead to false positive differential results. Too few cells means that the smear is too thin, which makes it difficult to observe the slide and may lead to false negative differential results. Heat fixing prevents a specimen from washing off the slide. It inactivates any enzymes present and prevents the cell from undergoing autolysis. It also kills the organism. To perform a heat fixing, hold the slide with a clothespin. Apply heat until the liquid evaporates. Basic and acidic dyes. Stains or dyes contain salts made of positive ion or a negative ion. Depending on the type of dye, the positive or negative ion may be chromophore, the colored ion, or the other uncolored ion called the counter ion. If the chromophore is positively charged, the stain is classified as a basic dye. If the negative ion is a chromophore, the stain is considered as an acidic dye. Positive and negative stains. Dyes are selected for staining based on the chemical properties of the dye and the specimen being observed. A positive stain is a dye that would be absorbed by the cells or organism being observed, adding color to objects of interest to make them stand out against the background. A negative stain is absorbed by the background, but not by the cells or organisms. Negative staining procedures produces an outline or a silhouette of the organism against a colorful background. The different types of staining procedures we will be talking about today is simple staining, negative staining, gram staining, acid-fast staining, endospore staining, capsule staining, and flagella staining. Simple staining. Simple stains paint the cells using a basic stain. It uses basic stains such as methanol blue, saffron, or crystal violet. A single dye is used to emphasize particular structures in the specimen. A simple stain will generally make all the organisms in the sample appear to be the same color, even if the sample contains more than one type of organism. To perform a simple stain, allow each slide to dry for approximately 1 minute and then heat fix the smears by holding the slide with a clothespin and placing the slide on a warm hot plate. Hold the slide on the hot plate only long enough for the liquid to evaporate. Place the slide on the staining rack and flood the slide with either saffron or crystal violet. Allow the stain stand for 1 minute and then rinse slowly with tap water. Carefully blot dry, but do not wipe the slide because this may remove the cells. Negative staining. Negative stains stain with acidic dye, India ink, and it colors the background of the slide. Negative stains uses acidic stains such as eosin, mangasin, or India ink. Most simple negative stains allow for visualization of cell morphology. To complete a negative stain, obtain 2 new clean microscopic slides, place 1 drop of India ink on one end of one slide, obtain an inoculum from your plated specimen, and emulsify the cells in the India ink. Using a second slide, drag the ink or bacteria mixture across the top of the first slide. Allow the slides to air dry completely, do not heat fix. Differential staining. Differential staining distinguishes organisms based on their interaction with multiple stains. Differential staining techniques commonly used in clinical settings include gram staining, acid-fast staining, endospore staining, flagella staining, and capsule staining. The primary stain is the first stain, colors all the cells. The mordant augments binding of the primary stain. Decolorizer removes primary stain selectively based on the presence or absence of specific characteristics. The counterstain is the contrasting color to the primary stain and stains the decolorized cells. Gram staining. The gram stain procedure is a differential staining procedure that involves multiple steps. First, crystal violet, a primary stain, is applied to the heat-fixed smear, giving all the cells a purple color. Next, gram iodine, a mordant, is added. A mordant is a substance used to stabilize stains or dyes. Gram iodine binds to the cell wall and forms crystal violet iodine complex. Next, a decolorizing agent is added. Cells that have a thick peptidoglycan layer in their cell walls are much less affected by the colorizing agent. However, the colorizing agent more easily washes the dye out of cells with thinner peptidoglycan layers, making them, again, colorless. Finally, a secondary counterstain, usually saffron, is added. This stains the decolorized cells pink and is less noticeable in cells that still contain crystal violet dye. The purple crystal violet stained cells are referred to as gram-positive cells, while the red saffron dyed cells are gram-negative. To perform a gram stain, prepare a bacterial smear for each species, then allow the slides to dry for approximately 1 minute and then heat fix the smears. Place the slide on the staining rack and flood with the primary stain, crystal violet. Allow the stain stand for 1 minute and then rinse slowly with tap water. Flood smear with the mordant, gram iodine, wait 1 minute and then rinse with tap water. Colorize by adding 95% ethanol while holding the slide at a 45-degree angle. Continue until no more purple stain runs off. Immediately rinse with tap water. Counterstain by flooding the slide with saffron for 1 minute. Rinse with tap water. Carefully blot dry. Acid-fast staining. An acid-fast stain is able to differentiate between two types of gram-positive cells, those that have a waxing mycolic acid in their cell wall and those that do not. Acid-fast staining is specific for the genus Mycobacterium. The primary stain used is carbofuscin. The mordant uses heat, which drives stain into cells. The decolorizer uses acid alcohol, which only removes color from the non-acid fast cells. The counterstain uses brilliant green, which stains the decolorized cells a tealish bluish green color. To perform an acid-fast staining, allow the slides to dry for approximately 1 minute and then heat fix the smear. Place the slides on the wire screen above the boiling water in the beaker on the hot plate and flood with carbofuscin. Heat for 3 minutes with the stain on the slide. Rinse thoroughly. Place the slide Decolorize by adding acid alcohol while holding the slides at a 45-degree angle. Continue until no more purple stain runs off. Immediately rinse with tap water. Place the slides on the wire screen above the boiling water in the beaker on the hot plate and flood with the counterstain brilliant green. Heat for 1 minute with the stain on the slide. Rinse with tap water and carefully blot dry. Endospore staining. The primary stain used is malachite green. The mordant uses heat, which dries stain to the spore, which helps penetrate the cortex layer. The decolorizer used is water. Malachite green is water soluble, and it only remains if trapped inside thick cortex layer of the endospore. The counterstain used is saffron, which stains vegetative cells and sporangia, not endospores. To perform an endospore staining, allow the slides to dry for approximately 1 minute and then heat fix the smear. Place the slides on the staining rack and flood with malachite green. Heat for 5 minutes by placing the slide on the wire screen above the boiling water in the beaker of the hot plate. Capsule staining. Capsule staining stained with acidic dye, India ink. Capsule stains retain neither acidic nor basic stains and have no ionic charge. The cell is stained with the basic dye and the background stained with acidic dye. Non-stained capsules appear white with the halo surrounding the cell. First air dry and gently rinse, and then stain with the basic dye crystal violet, then air dry and gently blot. Flagella staining. Flagella typically cannot be seen under a light microscope without specialized flagella staining technique. Flagella staining thickens the flagella by first applying the mordant, which coats the flagella. Then the specimen is stained with pyrosaline or basic fuchsin. The flagella staining is uncommon in clinical setting. The technique is commonly used by microbiologists. Conclusion. Staining microscopic specimens is important because it makes it easier to view the specimen under the microscope. Staining allows us to see the cell morphology, cell wall structures, and help identify unknown microorganisms. We discussed the different types of staining, which are simple staining, negative staining, gram staining, acid-fast staining, endospore staining, capsule staining, and flagella staining. Through my research, I've learned how different each staining procedure is and how it helps identify unknown microorganisms. This presentation should also help you learn how to do each staining procedure step by step to find out what specimen you have.